Okay, so I want to start off with this question I want you to think about. So how can you study something that's too small to see? So think DNA. If we were to look in your cells, could we just see your DNA? Probably not. It's too tight. So how do we study? How do we know what all the parts of DNA are? So can you think of any examples or any evidence of like how do we study something small? So before you say something out loud, I want you to take like 20 seconds, turn to the person next to you. If you're kind of the odd one out, make a group of 3. And just with a small group next to you, go over. How do we know how we study really tiny things? Okay, what kind of ideas do we have? Okay, so we have tools like microscopes that help us see. Do you all remember back at the beginning of the year, I showed you that video of all the different types of microscopes. So we have some that like we can see that can magnify up to like 1,000 times, and then there's even like those electron microscopes that can magnify up to like 500,000, or I think one can even go 1,000,000 times magnification. So those are huge. Can you think of anything else that's too tiny to see? Okay, like maybe like single cell. We could see single cells. What about just like what makes up DNA, right? What is this that we were just working with? Like molecules or atoms. Can we still see atoms on microscopes? No. Okay, so those are too small to see. Do we know atoms exist though? Yeah, we know atoms exist. How do we know things as small as atoms exist? What do you think? Did you say logic? Okay, like, can we also see like the effects of how they behave? Like, yeah. We've done experiments over time where we shoot things like X-rays at them. Can we see like an X-ray would like hit an atom and bounce off? Okay, so we have tools that either help us see really small things, or we still have techniques to get around and still see like the evidence of small things. All right, so now a second question I have is, what do you think the molecules of heredity are? So if you think back to Mendel times, so Mendel is the pea plant guy. He was the one that recognized that pea plants could pass down traits, or traits were inherited. It wasn't just a blend. And so after Mendel, so in like the early 1900s, scientists had a lot of questions of, what are the molecules that carry that heredity? So think back, what are just those macromolecules that we talked about at the beginning of the Earth? Those 4 main macromolecules that are inside of you. Okay, lipids are one of them. What's another one? Proteins, carbohydrates, and and we call that nucleic acids. Okay. So do you remember those 4 macromolecules? Mm-hmm. So in the 1900s, we knew about these 4 macromolecules. So we knew the unit of heredity had to be passed in one of those. Okay. Odds are scientists could rule out like lipids and carbs. They're just not big enough. So we could really narrow it down to proteins or nucleic acids, but we weren't quite sure which one. So starting at 1928 is when a handful of experiments were done. So we're going to go through those experiments. These should look familiar to the timeline that you did yesterday, but today I just want to go through them a little bit more in depth. So the first one is Griffiths Experiment. He published his results in 1928. And so he did this experiment with different strains of bacterial pneumonia and mice. Okay. So he had this virulent strain of pneumonia. Okay. It was called the S strain, and it caused the mouse to get sick and the mouse to die. He also had a non-virulent strain of pneumonia, which means the mouse wouldn't get sick. Therefore, when he injected it in the mouse lived. Now, at the time of his experiment, it was already well known that if you heated your substances, think of like your food, it would kill a lot of like harmful bacteria. So then he heated up the virulent strain of bacteria, so he killed any like harmful like substances in it. So then he injected it in the mouse, and just as he suspected, the mouse lived. So then he decided to go one step further. He took the heat-killed virulent strain, so the harmful strain, and he mixed it with the A virulent strain, or the non-harmful strain. And to his surprise, the mouse ended up dying. So that just tells him that somehow there had to be this transforming material that moved from the heat-killed strain to the A virulent strain. And so this is all the evidence Griffith had to actually show what he did. But in his mind, he reasoned that proteins are destroyed by heat. Therefore, DNA, or your nucleic acids, had to hold the genetic material. So that was the big conclusion, is between proteins or DNA, Griffith picked he picked DNA because proteins are destroyed by heat. Now, does he have any solid evidence that it's DNA and not proteins being passed back and forth between these two strains? Or is it just he just used his reasoning? Okay, so the scientific community is extremely skeptical. So do you think this is enough evidence to get the whole scientific community on board? Okay, it's not enough evidence. So he did this great experiment. He had some really good reasoning, but the scientific community is, well, you have to still prove it. This proves some sort of genetic material is being passed, but it doesn't actually narrow down DNA or protein. The story continues. So Avery continues the same experiment. This time it happens in 1944. And so he keeps the same experiment as Griffith. So he's still using the same strains of pneumonia, and he's still injecting it into mice. But now he's just trying to narrow down which macromolecule plays a role in it. So here he takes the non-virulent strain and the virulent strain that's been heated, and then he adds something called protease to it. Do you see how protease has almost part of protein in it? Okay. So by adding the protease, it makes it so proteins don't work in your body. So then the mouse dies, which is exactly what happened during Griffiths' experiment. So that means proteins have nothing to do with this genetic information being passed back and forth. So then he does the same experiment. This time he adds DNA-ase. DNA-ase, what do you think that inhibits? DNA. So this one makes it so DNA doesn't function. And this time, the mouse lives. So his results are the DNA-ase makes it so the DNA doesn't work. And since the mouse ended up living, unlike all the previous trials, it means that DNA has to be the holder of genetic information. You with me there? Okay, so his experiment got a lot more people on board. But still, is there any proof that you can actually see DNAs being passed back and forth between these strains? No. Okay, so more people in the scientific community on board, but not everyone. Some people want more proof. They want to see that it's actually DNA. So that brings us to our next experiment by two scientists with the last name Hershey and Chase. Okay, they published their study in 1952. They used something called a bacteriophage, which is this creepy looking virus. Okay, so viruses, they can't reproduce on their own. They have to take over a cell and they inject their genetic material into that cell. And so Hershey and Chase are going to use this bacteriophage to figure out if it's protein or DNA. All right, so I want to go through their experiment with you. I'm just going to zoom in just a tiny bit. Okay. So in Hershey and Chase's experiment, they use this bacteriophage, and they know that there's two main parts of a virus. You have proteins and you have DNA. Okay? And at this point in time, we know one of those holds genetic material. Okay, so they're going to figure out, is it the protein or is it the DNA? So they take radioactive elements and they label either the protein or they label the DNA with them. So in this first experiment, they labeled the protein. And so if you kind of see, they're showing the radioactivity by just highlighting it green. So the protein's green. They know that the bacteriophages are going to infect bacteria. So they're going to inject their genetic material into that bacteria. So then afterwards, when they study the bacteria, there's no radioactivity in the cell. Okay. Now, the only radioactive part is the protein. So did the protein make it into the cell? Okay, nope. So then they did a similar experiment. This time they had the bacteriophage. This time they added the radioactive elements to the DNA portion. So now when the bacteriophages inject their genetic info into the bacteria, this time when we look at it, it is radioactive. So what does that tell you got injected into the cell? DNA. So this was like the hard evidence the scientific community needed to see that DNA is what holds your genetic material. So now after that experiment, pretty much the scientific community was pretty on board of accepting that DNA is the molecule that holds your genetic info. However, there's still questions that remain. So a couple questions that remain was, how do nucleotides actually form DNA? And then how does DNA actually communicate the information that makes you you? So that's kind of what leads us to the next part of science. So we're just going to start with nucleotides. So nucleotides are like the base or foundation of nucleic acids. So nucleotides, they're composed of a 5-carbon sugar. It'll either be deoxyribose or ribose, depending on if we're talking about DNA or RNA. And we'll talk about that in the next couple slides. There's a phosphate group, and then one of 4 nitrogenous bases. And those are the bases we worked with at the beginning of class. So it can either be guanine or adenine, cytosine. In DNA, it's going to be thymine. And in RNA, it's going to be uracil. And I have those in upcoming slides, so we'll write those in a couple slides. So here's just like a generic image of a nucleotide. I think there's space in your notes to like roughly copy this down. Okay. So in your notes, copy down, there's the 5-carbon sugar. It's like this pentagon shape. Okay, so draw your little pentagon. Then coming up of it, do you see how you have like this right angle line? Make sure you copy this line. And then you can just make a circle here. You don't have to have like the P and the O's for the phosphate group. You can just draw a circle and label it phosphate group. And then on the other side of the sugar, you have this nitrogenous base. And you can just draw like a hexagon and just call it nitrogenous base. So go ahead and copy like the rough structure in your notes. All right, so nucleic acids are just made up of nucleotides. So nucleic acids are polymers. Does that word sound familiar? Hopefully from like the very beginning of the year. Nucleotides are monomers. So remember, monomers are just like single individual molecules. So nucleotides are going to bond together to make this really long chain, which is going to make the nucleic acid. Okay. And so in living cells, there's 2 types of nucleic acids. The first one is DNA, and the second one is RNA. So DNA stands for deoxyribonucleic acid. Go ahead and write that in your notes. So deoxyribonucleic acid. And it's still going to follow the same order of the nucleotide. So there's still going to be the 5-carbon sugar. This time it's deoxyribose. There's still a phosphate group. And then there'll still be one of the 4 nitrogenous bases. When it's DNA, the nitrogenous bases are adenine, guanine, cytosine, and thymine. RNA stands for ribonucleic acid. So make sure you get the ribonucleic acid in your notes. And this too follows the same structure of the nucleotide. So you still have the 5-carbon sugar. This time it's ribose. There's still a phosphate group, and then there's still one of 4 nitrogenous bases. In RNA, there's still adenine, still guanine, still cytosine, but this time there's uracil. So what base is missing from DNA? Thymine. Okay, so then wherever like a thymine would go in DNA, uracil takes its spot. All right, so when we look at our nitrogenous bases, we divide them up into 2 categories called purines and pyrimidines. Which one comes first on your notes? Okay, purines. So the purines are down here. When you just look at the shape, how does this shape differ than the shapes up here? Look at like their rings. We call the hexagons rings. How many rings are down here? 2. Okay. So purines have 2 rings. So go ahead and write that in your notes. And then also just list the purines. If you're getting tired of writing guanine and adenine, you can just write G and A. In your notes, you should have like a little blank for purines. So know that these are the ones with 2 rings and just G and A. From here on out, you're going to get familiar that G is always going to mean guanine, A is always going to mean adenine. And then our second group are pyrimidines. If you look at the shape up here, how many rings do they have? Okay, just 1 ring. We call like one of these a ring. Okay, so 1 ring. And then for the bases that are pyrimidines, it's cytosine, which you can just label C, uracil, which is U, and thymine, which is T. So if you want, from here on out into your notes, if you just want to abbreviate the base pairs by their first letter, that's totally fine. That's standard when it comes to talking about DNA, because how many times do you want to write out these words? Not many. So once we kind of get familiar that G stands guanine, you'll just start to write G. All right, so now in the mid-1900s, people are super interested in DNA, like in the scientific community. So now that we know DNA holds genetic material, we want to know, well, what's the structure of DNA? How does it actually look? How does it fit together? So Chargaff is the next scientist, and he was just comparing the amounts of adenine and guanine and thymine and cytosine in DNA samples. And he found that the amount of adenine always equals the amount of thymine, and then the amount of cytosine always equals the amount of guanine. So he came up with the conclusion that adenine is going to pair up with thymine, and guanine is going to pair up with cytosine. Or a purine is always going to bond to a pyrimidine. So make sure in your notes you have like A bonds to T, G bonds to C, which should be the same conclusion we came up with at the beginning of class, yes? Good. So now we still have the question of, well, what is the structure of DNA? Meaning, what does DNA actually look like? So 4 people were set out to try to answer this question, probably more than these 4, but these 4 made some big contributions of how we know what DNA looks like. So those 4 people were Rosalind Franklin, Maurice Wilkins, Francis Crick, and James Watson. So Rosalind Franklin, she was interested in figuring out what does DNA look like, so what's the structure. And so DNA is small, right? We can't see it. She used a technique called X-ray crystallography. And so she would send X-rays at DNA and look at what pattern they would make. And so this is her famous photo. And it took her a year to analyze this photo and figure out like what it means. Okay. So what she figured out from this photo is DNA, it's wide, it's tightly coiled, and it has this spiral shape to it. Okay. At the same time as Rosalind Franklin was working on this, other scientists were also working on this. Stole this picture without Rosalind Franklin knowing, and he gave it to Watson and Crick. 2 of them are Francis Crick and James Watson. They too were working on the structure of DNA and trying to figure out how everything fits together. And they were kind of missing this like final piece to the puzzle. Well, Maurice Wilkins, so then by looking at her picture showing that DNA is like wide and it's this spiral structure, Francis and Crick were able to finally like kind of tie it all together and solve the puzzle, if you will. So they're the ones that came up with the like formal official double helix of DNA. And so DNA's shape is a double helix, which just means like there's 2 spirals to it. If you think of it, I'm going to go back a slide. So then we relate the structure of DNA to like that spiral ladder shape. So the sides of the ladder are what we call the sugar phosphate backbone. And it's a backbone because it's strong, it's sturdy, everything attaches to it. Okay, so all those sugars and the phosphates that we talked about in the nucleotides, that makes up the backbone or like the sides of the ladder. And then those nitrogenous bases like adenine, thymine, cytosine, guanine, those face the interior together, and those would make like the rungs of a ladder or the steps. All right, so here's just another picture. So I like this more computer-generated picture. So again, here, this is what we call the sugar phosphate backbone. Okay, so this is where the sugars and the phosphates are. If you look at the actual, this is like the molecule model, so you can see like a lot of the reds and oranges. Okay, so that's our sugar phosphate background or backbone. And then on the inside, this is where your base pairs form. Okay, so you can see we have like the adenines, the thymines, guanines, cytosines. And do you see the little dotted lines here? Do you see like the little red? So these get held together by hydrogen bonds. And so that's going to help give DNA some of its properties that allow it to bond together, but it's also going to be able to be easily broken. And we'll talk about that when we talk about replication starting tomorrow. So again, here's just another picture of the structure of DNA. DNA is spiraled, but here's just like a flat picture so you can see it. So again, the sides of the ladder are going to be like sugar and phosphates, then your base is in the middle. So this is more like ladder looking, and then here's what it actually looks like. So it's really a spiral structure. And so one thing we know about the structure of DNA is the orientation of these strands. Okay, so one of them runs in this direction. We call it 5' to 3'. Over the next week or so, we'll talk more about what does that mean. Okay, so in case you're wondering what it means, we'll get there. It means how the carbons are labeled. But right, 5' to 3'. And then the other strain runs 3' to 5', which means they're anti-parallel. Okay, so think like one runs down, the other one runs up. And then the very last thing I want to talk about today is just difference between prokaryotes and eukaryotes. Okay, so they both have DNA. It's just a little bit different. So prokaryotes, DNA is just going to be in the cytoplasm. Remember, these don't have those like membrane-bound organelles. And you just get a ring of DNA and proteins in the prokaryote. And eukaryotes, these are our more complex cells. They have all the membrane-bound organelles, the ones that we have. DNA is organized into the individual chromosomes. That spiral, it tightens up and it forms our chromosomes. How many chromosomes do we have? 46. Okay, so our DNA gets like tightened up and condensed into all 46 of our chromosomes. Just kind of like a fun fact of the size and how many we have. The length of our human chromosomes, they range anywhere from 51 to 245 million base pairs. Okay. So that's how many like base pairs long our DNA is. And just to give you like a size reference, for every 140 million nucleotides, that would stretch out to be like 5 centimeters long. So when it's in our cells, that has to like really compact to fit in your cell.